everyone. Welcome to this first session of this afternoon. Let's start right away because you know that we have to be very careful about the time today. I remind this, the speakers already know. I will speak about this later on. Let me first thank the organizers of this wonderful meeting, and particularly Tom Rashpandari, who invited me to share this session, and thus giving me the opportunity to address you a few words. Here in this session, only for the sake of the high standard scientific presentation that we are seeing, it will be worth coming to this, attending to this meeting. But there is a very important goal, emotional goal of the meeting, which is to honor the dedication to science and mentoring of Dr. Khorana. It is actually this second element who decided me to come right away and join you on this special occasion to express my gratitude to Gobind for his teaching and accepting me in his lab for postdoctoral work. Everyone knows Gobind is a respected scientist worldwide and also sincerely acknowledged by all of the people who had the opportunity of working with him at that point in his long scientific career. In my case, Gobind accepted me in his lab and he taught me not only science but some other specific things, like for example when he used to say that we should make the laboratory our religion. Yesterday Peter King was also mentioning something about staying in the lab and not getting distracted from experimental work. So these were very important lessons Gobind taught me, and of course he also insisted that we should work in the lab as long as we had some energy to do so. In the modern times we are living, where everything is moving very fast, sometimes even too fast, the Gobind's vision on science still stands solid as a rock, but it is arguably too demanding for many people. But still, I believe that it's a very important and good example especially for young scientists. In this meeting, we have seen and we will continue to see many different scientific topics being covered. And what is interesting to note is that many of these topics have, you can find Gobind's footsteps and inspiration in all of this work. In going from the nucleic acids work to membrane proteins, from genetic code studies to signal transduction mediated by membrane receptors, and in the last part of his career, all of his important work on bacteriorhodopsin, elucidating the mechanism of proton transport across the membranes, the bacterial membranes, and of course, the work I was involved with also, that is the visual transduction, phototransduction, the important contribution in structure function studies of rhodopsin and other proteins of the visual cascade like arrestin, transducin, and rhodopsin kinase. I have said these few words about Gobind, knowing that it's very difficult to be original in saying something about him, given his immense scientific merit, and that many of you know him better and have worked for longer times with him than I have. But I decided to say this because I felt I should, and also because this session is given by a number of speakers, most of them corresponding to the MIT period of life of the scientific life of Gobind. In this case, that was my case particularly. I joined MIT and I spent there some years as a postdoctoral fellow, later on as visiting scientist, and starting in building 18, which was the chemistry building, and then going to the new building. I participated in the move to the new biology building, actually with with one of the persons that is going to give a talk today, the parents who's in the audience. I remember going to the basement corridors of MIT from the chemistry to the biology building with our small freezers and our precious samples, trying not to lose anything. And another thing I remember, another anecdote of one of the presenting speakers, is that one of the first things I saw when I stepped into room 18524 of the chemistry building, I could read Tom Sagmar in one of the drawers of the lab in this room. Tom is one of the other speakers of today's session. And really, he preceded me in redoxing work and had already left when I joined Gordon's lab. So today's session, this session is given by 5 very important and outstanding researchers, and the first one would be Julius Adler, who is a professor in this host university, Wisconsin-Madison University, and he will speak about the current research he's doing on Drosophila behavior and the response of the flies to environmental signals. The 4 other speakers correspond to this MIT period of Gobind's life. 3 of them are former Gobind's alumni. One of them is Marialda Gilles Gonzalez, who did her PhD under Gobind on bacterial redoxing structure and function. The other 2 are the aforementioned Tom Sagmar and Dave Farns, who worked on redoxing in very important part. First Tom in the first studies on redoxing, and then Dave Farns took over by studying application of biophysical methods. Tom has been working with other receptors too, trying to find out differences in structure and function of these self-dependent receptors. And finally, the last speaker, which is not the last speaker chronologically, but the last person to mention is Wayne Howell, a very well-known and respected biophysicist who has collaborated with Gobind in the recent years in studying redoxing, structure, dynamics, particularly by EPR spectroscopy. So all these people are living examples, clear examples of people like others that we have seen and we continue to see in this meeting, of people that have learned from Gobind at different stages of their life, at the predoctoral and postdoctoral level, collaborating with him in interesting projects, and also by reading his rigorous and very important bibliography on all the subjects that he has approached. So, without any other delay, I just give my microphone to... Well, I should say... Yeah, I should say, remind the speaker, maybe I hope I was. I didn't understand what I did. Sorry, I was doing very hard to be the chairman and to keep people.
I share a long history with some of the people who are here today. It was 55 years ago that I was a graduate student of Henry Lark, working on metabolism of some fatty acids like propionate and others in rat liver mitochondria. Hank was a great inspiration to me, one of the greatest in my life. And I admire the patience that he had with all of his graduate students and the enthusiasm that he had for all of their work. "50 years ago, after doing a postdoc postdoc with Arthur Kornberg at Stanford and Dale Kaiser at Stanford. I met Marshall Nuremberg at Cold Spring Harbor. Marshall and I took together the bacterial genetics course. And he became a very good friend of mine. He told me about the dreams that he had about studying the coding problem. And I told him about the dreams that I had about studying behavior. Then it was 49 years ago that I came here. Gobind came here almost the same day. As I told some of you yesterday, Gobind was taking a hike out on Picnic Point, and I happened also to be taking a hike out on Picnic Point. And I said to Gobind, "Are you Gobind Khorana?" "I'm Julius Adler." And ever since that day, Gobind and I have been the greatest of friends. I worked part-time with Gobind the first year that I was here. Together, he and I and Arturo Falaschi, who may not be here, did a paper on RNA synthesis directed by DNA of various kinds. And so I'm very pleased and honored to be back here with Gobind today. This is my talk for today. It's a paper that I'm submitting to Annual Review of Biochemistry for next year. It is now in a rough draft, and here are copies if any of you would like to criticize it. But seriously, I would really like to share with you your thoughts and your severe criticism of these ideas. In search of the boss, the thing that is in charge of each organism. This is an experiment that we've been doing the past few years. Here we isolated mutants in the following way. This is a long, big tube. Drosophila flies, about 500 of them that were mutagenized are put here. And then here, I share an interest with Gobind. Namely, we're studying light, as Gobind did at MIT, with insect vision and animal vision. So there's a gradient of light put in here, and there's a gradient of a repulsive odor that is put in here. The flies have to decide what to do, whether to be attracted by this light or to be repelled by that odor. What they decide depends on how much light there is and how much odor there is. If there's just a little more odor than there is light, then the flies all stay here. But among them, among these mutagenized flies, are a very few that don't know how to do that. So we reach in and take out these very few and study them further to see what they are. This is an assay measuring attraction by the light and repulsion by the odor. There is light all the time here. At zero odor, there's no odor. And the flies, the wild type, the parental flies, are largely attracted. Then as the odor concentration gets higher and higher and higher, the normal wild-type flies are repelled. But there are mutants here that are not repelled at these high concentrations of repellent. These mutants move more or less randomly. 33 1/3% on this scale would be random movement. And these are the mutants that we wanted to study and have studied. So among these mutants are some that at 34 degrees, at this high concentration of repellent, don't know what to do and just move randomly, whereas their parent is repelled, as you saw in that figure. If you give them light alone, the mutants are attracted just like the parents are attracted. If you give them repellent alone, the mutants are repelled just as the parents are repelled. So it's only when the light is together with the repellent that the mutants have a problem. This problem arises at 34 degrees. At room temperature, these mutants are normal. So here it is at room temperature. They're repelled just like the parents are repelled. And they're attracted like the parents are attracted, and they're repelled like the parents are repelled. So the conclusion is that we have found mutants that are temperature-dependent mutants. They have a problem with 34, not at 22, for light plus odor. But they're normal for light alone, and they're normal for odor alone. So we're currently working on these mutants. We've isolated 9 of them, starting out with about 1,000 or more mutagenetic flies. And we'd like to get maybe 100 of those if possible. We'd like to find out if the mutation is only for a certain intensity of light and a certain concentration of odor. So we'd like to vary the strength of the light and vary the concentration of the odor further than what's shown here. We'd like to know if the mutation is specific for attractive light and repulsive odor, or instead, could one add other attractants and other repellents, for example, an attractive odor and repulsive heat. Then we'd like to find out how many genes are involved, and we'd like to map these mutants to find out where these genes map. And ultimately, we want to find out the function of those genes. What do those genes do? What does the normal form of those genes do to tell the creature, here is an attractive stimulus and a repulsive stimulus, you must do this or you must do that? And what is the mechanism for that action? And does that mechanism occur elsewhere among living creatures, such as mice or people or E. coli or what? The pathway in all these creatures in general is that there are external stimuli sensed by these ways, and there are internal stimuli sensed by these ways. The external stimuli come together and the internal stimuli come together, and then centrally there is something that coordinates this with that to bring about a response. We'd like to know the mechanism of what is shown here. We think that there's something which we like to call the vault, which controls that mechanism. When there is attractive and repulsive sensing of external stimuli, such as you saw in the slide, where there was light and repulsive smell. The boss might act here. Or the boss might act here if there are internal stimuli, some of them attractive and some of them repulsive. Or the boss might act here if there is an external stimulus and an internal stimulus. So we want to find out where these different mutants are. Some of them may be here, some of them may be here. And they are controlled by something that we call the boss. And we want to find mutants in the boss so that we can characterize this thing, find out what it is and how it works. That's our program. Things like this occur in mammals as well. The brain has a part to it shown in blue, which is the prefrontal cortex. In people, a large part of the brain is prefrontal cortex. In chimp less, in rhesus monkey less, in dog less, in cat less. In the invertebrates, we don't know if there is something like prefrontal cortex because the anatomy of the invertebrate brain is very different from that of the vertebrate brain, though functionally they are often very similar. This has been studied in people. Here's an experiment done by Rolls and his group at Oxford. On one side of the hand is put something warm. On the other side of the hand is something cold. And then the human subject has to decide whether he liked this hot thing or this hot, comfortable thing or this cold, uncomfortable thing. And then, a few seconds later, they have to declare the decision. This is all fed into functional magnetic resonance imaging, and it records the brain responses. The result from this group is that the response occurs in the prefrontal cortex. They've done a similar and more recent experiment, 2009, using, rather than warm and cold, they've been using an attractive smell together with a repulsive smell and come to the same conclusion that it is in the prefrontal cortex that the decision gets made. So I like the idea that behavior is ultimately controlled by some essential gene, which in turn is controlled by the boss. Essential genes, let me define, are those genes that a creature must have in order to stay alive in a rich medium. Most of the genes of a creature, some 80% of them, can be removed by mutation and the creature is still alive. But these genes, called essential genes, must be there in order for life to occur. Presumably, all the kinds of behavior of creatures are controlled by essential genes in the body, like DNA synthesis or RNA synthesis, protein synthesis, development, and so on. This has been studied to a small degree. In E. coli. E. coli has 303 of these essential genes out of a total of around 4,000 genes. Only 303 of them are essential. And one of these essential genes controls the DNA polymerase, another controls RNA synthesis, protein synthesis, development, and so on. And we want to find out which of them controls behavior. Of these 303, there are 30 of them with still an unknown function. And we imagine that one or more of these are responsible for the control of behavior. This is work that has been carried out by Fred Blattner and his group. Fred is going to speak here. And by a Japanese group together with Warner. So my great interest in this is one is an essential gene for carrying out behavior, both in bacteria and in Drosophila and in people. And is there such a thing as the boss, which controls these essential genes? This slide taken from these people, I don't know anything about it in detail. I show it to you only because it tells that for a certain single gene called P53, many, many, many things are ultimately controlled. So it's possible to find single genes that control many things, and I imagine it might be possible to find the boss which controls all of those genes. Here's an example of what I have in mind. This is work carried out by these people, and it's in E. coli. At the very top are what are called global regulators. These are responsible for everything below. Next are major regulators, responsible for everything below, minor regulators, and then the target genes. This kind of conclusion has been reached for maybe a 20th or a 10th of all of the E. coli genes, and needs to be amplified, which according to the authors may take a few years, so that we can find out what the whole picture looks like and find out what perhaps the boss is on top of